Hello, everybody. My name is Rogelio Hernandez Lopez, and I'm an assistant professor in the departments of bioengineering and genetics at Stanford University. Um, Today, I'm really glad to be here, so I'm going to tell you a little bit about my academic trajectory and the research that I did as a grad student and as a postdoc, and now happening in my lab. But because I guess the number of people is small enough, let's make this interactive. So if you have any question at any point, please feel free to either put it in the chat or just open your mic and maybe raise your hand, ask me a question. No, yeah. So today I'm gonna tell you about three different things. I'm gonna start by discussing or telling you how I got into science and where I have lived and what I found interesting and cool about doing science and being a researcher now. Then I'm going to tell you a little bit about a couple of problems that got me interested during my PhD and my postdoctoral research. And I'm going to conclude by telling you about a couple of projects that are very close to my heart and that are related to STEM education, outreach, and the training of the next generation of scientists. Okay, so I am originally from Mexico, and I grew up in Oaxaca, which is a city in the south of Mexico. And for me, a very important moment in my life to decide to become a scientist was during high school, and in particular, the process of participating in the science Olympiads. You might be aware or not, but like science Olympiads are these events that actually are worldwide. So there is different stages of these contests at the kind of city and regional and national level, and then later on there is an international competition. And I started in mathematics, but during the process I also was aware that there exist other Olympiads like chemistry and biology and physics. I think now recently there is also Olympiads in computer science. So these activities, at least in my case, were great for learning about concepts and things that were beyond what you will learn in school. So that was something that I really enjoy, learning new things and solving problems. I also enjoyed that I met a group of other students and friends who were equally passionate about science. And I think just creating this community of students and learners that are very into the same topic was something that I enjoy a lot. So I had the opportunity to participate in the International Chemistry Olympiad in 2003. It was the first time I actually traveled outside Mexico. So I went to Athens right before the Olympic Games of 2004. And it was like a wonderful experience because yeah, just meeting other students around the world and like visiting a new city and traveling. So all for that was like a very transformative experience. And that's where I also decided that I wanted to be a scientist and that I wanted to continue studying science as a long-term career. So because I guess I got good grades and I was considered a good student in chemistry, I decided to pursue my major in chemistry. So I moved to Mexico City, and this picture that I am showing you here is my undergrad lab where I did research over a couple of years in the field of biochemistry. And I'm going to tell you in a moment a little bit more about that. But like something that I just want to point out is that starting from the first summer that I had in my first year of college, I started doing research. And so depending on what stage you are now in your education, I think if you are curious about what the academic life is or what the day-to-day of a researcher is, there is multiple programs most likely, maybe at your university or other universities nearby that can host undergrad students to do research. So I was fortunate that I had the opportunity to participate in these summer and winter programs. And so I was jumping through different fields to actually find what I liked. No, at the beginning I thought that I liked everything, no, but then through experience and once you go and do a little bit of research, then you will realize maybe that uh, some fields or day-to-days are different than others or harder than others or just like your preference style. So I decided to start moving into the direction of biochemistry at that moment. I was also very interested in physics and math. And at the end of my undergrad, I was actually auditing some classes from pure physics, pure math. So that's what drew me into, for grad school, applying to the chemical physics program. And I applied to a few universities in the US. I got admitted to Harvard, and I decided to pursue my PhD in chemical physics at Harvard. But something that maybe is uh, um, not everybody knows, no, and I didn't know, but at that time, but through friends and through other people and asking a lot of questions, I got to know that to go to grad school, they pay you, no, you don't have to pay. That there is a whole process, no, that you have to prepare in advance in terms of taking exams. At that time, like you had to take the GRE, and because I was an international student, you had to I had to take English tests. But there is so a whole process of preparing your statement of purpose and figuring out, who are the professors that you might want to work with. So it's a whole process that, like, the sooner you start, the better for the entire process. But so that was my case. I thought that I wanted to go into a field that was multidisciplinary, and I found in the Leshner lab at Harvard, who at that time was studying molecular motors, was the perfect fit for me because, as I'm going to show you in a few slides, it incorporates a little bit of like multiple fields. For example, chemistry, biology, physics, no, computational image processing. So I think I always was drawn to these fields that are multidisciplinary. And because of that, at the end of my PhD and in my transition to my postdoctoral research, I continue pursuing research in fields that I consider multidisciplinary. And I transitioned from working with proteins and trying to solve the structures and understand the mechanisms of proteins to working with cells, no? And I will tell you in a moment what specifically I mean by T-cell engineering and what can you do with synthetic biology. But I decided to move into this new field. Here's another picture of the lab of Wendell Lim, where I did my postdoctoral research. And I moved in actually 2015 to the Bay Area. And I was a postdoc for a few years before starting my lab at Stanford in 2022. So this picture now is maybe a couple of years old, but here is my research group. And I'll tell you in a moment more about what we are studying. So let me just now transition to tell you a very brief stories of two different research areas that I was fascinated and I continue to be fascinated during my career. So first, I'm gonna tell you something at the molecular scale. So, and then what I mean by that is like proteins, no, in the nanometer, hundreds of nanometers scale. And I'm gonna tell you in particular about this group of proteins that are called molecular motors. Then I'm going to tell you about the cellular scale and the work that I did during my postdoc very briefly, because we're going to have an opportunity to discuss more details in a few weeks about T-cell engineering and how you can use T-cells to kill cancer. And then I'm going to tell you just very briefly some of the questions that we're trying to answer in my lab. So I'm going to start with the molecular motors. So I want you to draw your attention to this picture. That is a beautiful picture of two cells. No? So here, what you see is two cells using a fluorescent microscope. And in purple, you have the actin filaments. In yellow, you see the microtubules. And in green, the nuclei of the cell. So if you zoom further into these cells, what you will find is that there is these tracks. No, these are roads for groups of proteins that are called molecular motors. And for example, myosins walk along actin filaments, these things in blue. And dyneins and kinesins move along the microtubule. So if you go and Google online, no, dynein, kinesin, I'm sure that you will find these beautiful movies of these molecular motors that are moving cargo around. So these are very important proteins in our cells to transport organelles, to move neurotransmitters. For example, they are very important for synapse formation and synapse communication in neuroscience. But one of the questions that I wanted to answer and understand more about was how these motors work at the molecular level. And also, I show you this movie and this cartoon of something that has a shape like a tiny human or a robot, no, that has two different units, and they move one after the other. But if you think about for a moment, this is all driven by chemical interactions, no? So this is all protein-protein interaction. So it's just fascinating to me to think about that there exist proteins that function and that we can think about, and we're trying to understand how is it that they kind of move in a way that kind of resembles, no, how we walk as a human being. So because of the shape of these proteins and because of there is two units, so usually people think about in the context of dynein as the foot of the dynein at the head of the dynein. So the head is the site where it catalyzes or burns through this molecule called ATP, which is the source of energy for the movement of these motors. And so one big question during my PhD that I tried to answer was, how is it that the foot communicates with the head? No, like again, think about how we walk as human beings. So we have a brain, and there is all these signals that need to happen in order for us to move around. But again, all these in the context of these molecular motors is happening at the molecular level and through protein-protein interaction. So it was just fascinating to me to think about this communication. So without making the story too long, I just want to mention that we use very fancy microscopes and some computational analysis to come up with the shape of this foot of the dynein. No, so this is many years of work summarized into one picture. But there is now very powerful microscope that allow you to look at the molecular level, at the even atomic resolution level. At the time, we didn't have that resolution, but now with the new methods, you can have even atomic resolution to look at what is the shape and the interactions that happen of this foot of the protein and its track, you know, the tubulin dimer or microtubules. We were able also to use molecular dynamic simulations to look at the interactions between that foot and the track, the microtubule, and later on use this very detailed information to test some predictions in the context of dimers or these molecules that move. So before I show you this movie, where you see these tiny dots that are moving around, you don't see along what they are moving on, but so we have very special ways in which we can label the dimers. So for example, these little dots here in red are cartoons to depict the fluorophores or these fluorescent molecules that we can use to label where these motors are. And in this case, the microtubules are not labeled, but you could also label them. And so we have ways in which you can also look at the movement of these motors and quantify how fast they move, how long they move, and to try to understand, how these chemical interactions that I'm showing you here in the movie to the left modulate the motility or the movement of these motors. And again, I am not gonna tell you the full story, but something that I want to mention is that because we had this very detailed information of the interactions, we were able to predict what will happen if we make mutations or substitutions into this protein. No, for example, there were mutations where we were changing one of these glutamate amino acids, like that are shown in as with the letter E, for a glutamine, or for example, a glutamate to a lysine. No, maybe you don't know the names of these residues, maybe you do, but essentially what I'm showing you is how we were able to engineer motors that now will walk many more times longer than the original dining motor. So the wild type version, so the version that it's known in nature, at least in the context of these motors in yeast, only walks about 2 microns. But with the mutations that we introduced and the measurements we did, we were able to create motors that now walk 10 microns or 12 microns. No, so that was something that kind of tells you how we use approaches from different fields and image processing, molecular dynamics, single molecule biophysics to understand the mechanism of these proteins and to engineer even versions that are very different. So there is one question here. Yes.
I was just curious, in that video that you're showing, you were saying like the little dots that are moving are the dyneins and they're moving along a microtubule. So can you not see that microtubule because it's much, much smaller, or is it because the dyneins are tagged with something?
It's because both. So the dyneins are tagged with fluorescent molecule. That's why we can see them. And in this case, we didn't label the microtubules. So in this particular movie, we didn't label the microtubules, but you could label the microtubules. And we have experiments where essentially when you polymerize or when you build these microtubules, you include fluorescently labeled tubulin so that now you can also see the track. And we often in the lab do those type of assays, no, where you label both or you only label one or you label the other, no? So depending on where you are trying to track. But yeah, that's a good question. So all these details, yeah.
Sorry, I was just curious. What is like the scale of those two? Like, are the dynein proteins much larger than the microtubules?
No, the microtubule is many microns. So it could be, for example, here I'm showing you that the length of these dynein motors, they walk for maybe 10 microns, 12 microns or more. And the size of these proteins is in the nanometer scale, no? It's like, so the analogy is that the microtubules are roads, no? So imagine just like a highway and the dynein motors are cars, no? Like they're much smaller than, that's why you see here, this movie, you don't see the microtubule, but you see all this length, no, at least, and then you see there is like 10 or more of these motors that can move at the same time along that microtubule.
Got it. Okay, thank you.
Yeah, so let me quickly now tell you a little bit about the cellular scale and another problem that I'm really fascinated about, which is now how we can engineer T cells to kill cancer. So, and by the way, in this movie, which was collected by one of my friends, Alex Ritter, what you're showing is a T cell that is encountering a leukemia cancer cell, and at some point, you don't see it here, but is deciding to start killing it. But like, at least you see the first interactions where the T cell in orange is detecting the cancer cell in blue. So, we are in the lab very fascinated about the idea of using our own immune cells as a killer cell that can now be retargeted to kill tumor cells. So here what I'm showing you is a spheroid, a tumor spheroid that we can create in the lab in green, that is made of many cancer cells. You can actually see maybe that are like the outlines of a bunch of cells. And in orange, what you see are T cells. So thanks to the research of many groups around the world, now we know how to engineer these T cells to direct them so that they can now identify, migrate, infiltrate, and kill tumor cells. So I'm going to tell you a little bit of more details on how this is done, but I just want you to give the idea that now it's possible to engineer T cells with synthetic receptors that allow to direct this killing activity that already T cells have, but now against cancer cells. And the idea is that you engineer these T cells with new synthetic receptors. And these synthetic receptors allow the T cells to find proteins that are expressed on the surface of cancer cells. And in that way, if these T cells encounter those target cells, then they will start killing them. This type of approach is known as immunotherapy, and in particular, chimeric antigen receptor T cells are these T cells that are modified with these synthetic receptors. And this approach has worked very successfully in the context of blood cancers, in lymphoma, leukemia, where the protein that these T cells are identifying is the protein known as CD19. There is new versions or other versions of these therapies that are recognizing the protein CD20 and CD22, BCMA. So the names are not important, but what is important is that there is now different flavors of these therapies that target different antigens or proteins. And in fact, up to date now, there is 6 FDA-approved therapies of these chimeric antigen receptors that are known to have good efficacy in the context of blood cancers. So in my lab, we're interested in extending these approaches and using this type of technologies and ideas, but for the context of solid tumors, for example, breast cancer, lung cancer, ovarian cancer, no, where there is a few other challenges, and where in addition to these chimeric receptors, we need other tools so that we can safely and efficiently target these cancers. So one of the topics that I will discuss in a few weeks is more details about this new synthetic circuit that I devised and designed during my postdoc to direct the activity of T cells against only tumors without killing other cells that may also have these proteins, but that are normal tissue. So, which is one of the challenges in the context of cancers like HER2 positive breast cancer, or a few versions of lung cancer, where there is tissues that are normal. For example, your lungs, your skin, your heart have low levels of these proteins, and but cancer has a bunch of these proteins. So the amount of protein that is expressed on the surface of cancer cells each is maybe 100 more times larger than the amount that you will find in other tissues. So if you use a regular CAR T cell, as the one that I showed you in the previous movie, what you will have is toxicity, because now those T cells, the traditional T cells, will kill both types of targets, regardless of how much protein there is on their surface. What I'm proposing, and what I'm going to tell you more in a few weeks, is a way to now control the function of these T cells so that you only kill the ones that have a very large amount of protein, but spare the cells that have low levels of the antigen. Yes, there is a question.
We have some questions in the chat. We have
Oh,
a few questions.
Okay, it says here. How could you make killer? I have a question. How could you make a killer cell attack the cancer cells due to the cancer cell have PD-L1 that bind with PD-1 and inhibition to the activity of T cell? Okay, let me see the other question. Can you briefly explain or describe the mechanism regarding PDL2? This is a different. Are you making a mutation in the T cell that make it block the binding of the ligand to enable it attack cancer cell or what? Okay, so all these questions are very important. I could maybe in a few minutes go back to them and tell you a little bit more. And yes, so I'll let me continue to the next slide, and then I will go back to answer these questions. So I'm just to summarize some of the work that we are doing in my lab. We are interested in creating new ways in which T cells can target cancer cells, perhaps engineering novel behavior such as trafficking to the tumor, infiltrating into the tumors, the specific recognition of cancer cells while sparing normal tissues. We also use methods to try to mimic and model the tumor microenvironment and the spatial organization of these tumors. And we also use animal models of cancer to test in further the efficacy and safety of these therapies in primarily mouse models of cancer. And among with these categories, now there are studies that are at the level of protein-protein interaction. For example, the receptors that recognize the proteins, what is their binding affinity, what is their biochemistry. We use methods from genome engineering to introduce these synthetic receptors and synthetic circuits into the T cells, and we are trying to understand their dynamics. How long does it take a T cell to activate? Once it's activated, how long does it take to turn off? In the context of the tumor spheroids and the spatial organization of the tumors, we are trying to understand with different technologies how the signals that the tumors present affect the function of T cells. For example, a couple of questions in the chat are related to this PD-1, PD-L1 axis, which is a mechanism by which the tumor cells, for example, can counteract or inhibit the function of T cells, And so because we know that the tumor microenvironment actually is presenting and secreting signals that will dampen the function of T cells, then we have to design also strategies for the T cells to counteract that those types of mechanisms. And in the context of the mouse models, well, we are trying to also recapitulate either the microenvironment or the location of these tumors, and try to assess also their safety and efficacy. So just before concluding this part, we are located in the Clark Center, at Stanford University. This is a picture of my building, and we are actually here in the second floor. Here is my email, the website, my X handle. So let me answer a few other questions here in the chat before moving into the second part of science education. There is an interaction between the CAR T cell and killer cells that will be accounted for by the type of CAR or what generation of the CAR is. The CAR might stimulate other proteins that will assist the killer cells to recognize potential cells that are cancerous. So there is questions or comments here about, of course, the very simplistic way is that you are targeting T cells to recognize specific features of cancer cells. But that's not the whole story, no. As I said, the tumors are presenting different types of signals, no. Some of these signals could be inhibiting the function of T cells, and for example, the PD-1, PD-L1 axis is one. There is other exhaustion markers, LAG3, TIM3. There is transcription factors that get activated within the T cells. So we are pursuing several approaches that try to deal with all these different axes, no? So in some of the problems that we are trying to understand, our approaches that we're taking is seeing what happens if you downregulate or suppress the activity of multiple transcription factors that are known to suppress the function of T cells. So we are doing that through either genetic perturbations using CRISPR Cas9 interference or activation or knockouts, no, of multiple genes. We are also using different types of receptors that allow you to not only interact with tumor cells, but maybe also with other cells like dendritic cells, B cells, macrophages that are known to also be important for the anti-tumor activity in the immune system. So we are aware that not all the job is gonna get done by the T cells, and therefore we are engineering T cells to communicate or work together with other immune cells so that you can overcome this immunosuppressive tumor microenvironment. So yeah, so the problem of cancer immunotherapy and the approaches that should be taken to make these technologies widely applicable to different types of cancers will depend on what the cancer type is, and also the characteristics of the tumor microenvironment. So potentially or maybe multiple approaches are multiple circuits and perturbations are going to be needed to be able to deal with all these problems that engineered T cells face when dealing with solid tumors. Okay, so I'm going to move on now to second part so that we can leave at least a few more minutes to questions. So one thing that I found out also through the process of grad school and now later as a postdoc and now as a professor is that it's very important to contribute to the education of the public in general, but also to demystify, what science is, how you become a scientist, and to train the next generations of scientists and engineers around the world. And because of that, when I was a grad student and together with a few friends, we started a program that is called Clubes de Ciencia, which stands for Science Clubs in Spanish. So I'm going to tell you in a moment what the program looks like. So here I'm just showing you a few pictures. So it's a hands-on program where we offer workshops for high school and college students. And the goal is to make them aware, students to be aware about what the potential is to be in science and pursue a career in science. We are also trying to inspire them and show them that science is fun and cool, and innovations happen because of people like us, and like you that are interested in science and that are passionate about science. So I'm gonna play video that tries to describe a little bit more about the program and that captures some of the comments from some of the participants. So it's in Spanish, but it has like English subtitles, so hopefully this is gonna work.
Mi nombre es Manuel Razo Mejía, soy originario de León, Guanajuato, y actualmente me encuentro trabajando en la Universidad de Stanford, en California.
My name is Kiarash Chalmardani. I'm a rising fifth-year cancer biology PhD candidate at Stanford.
Soy Eric Leonel Espinosa Villatoro, de Chiapas. Actualmente estoy haciendo un postdoctorado en la Universidad de Stanford.
Hola, soy Josh. Yo he estudiado en doctorado en Stanford, en los Estados Unidos.
My name's Karen Malakun. I'm an MD PhD student in neuroscience at Stanford.
Clubes de Ciencia es una idea increíblemente sencilla, pero que puede transformar realmente la vida de la gente.
The clubs exist to be able to transfer knowledge to students, the next generation of the scientists, and be able to share what made me excited or what made us excited, that you're probably on average like 10 years older than them, and get them excited.
Y la idea es simplemente conectar a chavos y chavas que están interesados en la ciencia con científicos. Los estudiantes aplican para ciertos cursos que están diseñados por dos instructores, uno basado en México y uno basado en los Estados Unidos principalmente. El chiste es hacer como un curso donde no sea lo que viene en el libro de texto, sino que sea en las fronteras de lo que la humanidad sabe y no sabe.
I definitely learned a lot more from my students and from the other instructors, and I really want to encourage, anyone in high school or early college to take advantage of this great opportunity. You really are the future of this country and the world.
He disfrutado muchísimo para conocer a tanta gente tan listos y tan amables, y también para la oportunidad para enseñar a esos estudiantes increíbles que son muy listos y muy felices.
Soy orgullosamente mexicano, viviendo en otro país, pero queriendo siempre tener este tipo de plataformas como clubes de ciencia para que nosotros podamos seguir compartiendo conocimiento y nutriendo a la nueva generación para que ellos tengan el interés por hacer ciencia aquí y en el mundo.
We've been working with the amazing people here, not just the students, but also the staff and the coordinators and all the people and the other instructors. I got to know people who I probably would have never met, and made friendships that probably would last forever.
Okay, yeah, so the program, because it's a very simple idea, the program started in Mexico. Now it's been 10 years since the program started, but later it's expanded to Colombia and Peru and Brazil. And at some point, we also had some activities in Spain and Paraguay and Bolivia. With the pandemic, some of the programs in some countries kind of like downsized, but now we are trying to bring back all the teams and groups of instructors and coordinators that were previously operating. And so far, around more than 20,000 students have participated into this program. And we also learned during 2019, 2021, that we could combine digital education with hands-on education. So we also have some content that is online, that is freely available. So all this program is free. Students do not have to pay anything. Just if they get selected, they can participate. Okay. So just to summarize, today I told you a little bit about how I became a scientist and what drew me into science, what type of problems I'm interested. I just briefly touched on molecular motors and cell therapies for cancer. And I also mentioned a little bit about our work with STEM education and outreach in Latin America and Mexico in particular. This is a picture of my lab at the genetics retreat at Stanford last year. And as you can see, we have students from bioengineering, cancer biology, genetics. There is some students that are medical, MD students, and so on. So we're trying to build a multidisciplinary team to work on these type of problems. And yeah, with that, I'd like to maybe take a few other questions and just answer whatever you feel that could be helpful.
A couple more questions in the chat. By Nishat, the last question.
It says, are you exploring the role of fat cells in the tumor microenvironment in terms of helping the tumor cells survive? That's a good question. No, we are not looking into that in particular, but we are looking into metabolism. So for example, it's known that the core of tumors, and in fact, the tumor microenvironment is not only cancer cells. So there is different types of immune cells. There is a stroma. There is it's a microenvironment, no? So it's a community. So, and we're trying to model some aspects of it. So, for example, some of these 3D spheroids that I mentioned, sometimes we also include fibroblasts there, or we are trying to include extracellular matrix. We are trying to model the hypoxic conditions, so the low levels of oxygens, by using chambers that mimic those types of conditions. No, so the amounts of oxygens that are found in the tumors are very different from other tissues. So we're trying to understand what are the roles of different of these different elements into the functions of the cancer cells, but also on the T cells, no, and how we can engineer new ways to improve the function of T cells into these environments. It says here, interested in early diseases detection, utilizing AI innovations for improving African healthcare system. Okay, so thank you for this note. Yeah, we are in the space of therapeutics development. So of course, the field of cancer is huge, and we need many players, many different areas. There is a lot of work not trying to identify using artificial intelligence what are the subsets, no, of patients, no, that may respond better to this type of cell therapies, no, and definitely with machine learning AI, the vast number of data that we have now from clinical trials or the profiles of patients, no, we hope that we can find, these subsets and maybe design personalized therapies, or find also from the therapies we have who are the most likely patients that are going to respond. There is another question here, Leila.
Hi, so I had two questions, one pertaining to your organization. If we're kind of interested in helping out, or if we know someone, is it just very easy to go to the website and kind of sign up, or?
Yes. Yeah. So to participate in the program as a volunteer or as an instructor or as a student, yeah, all these happens online through the website. So we are always looking for more people to contribute, to participate. So to participate as an instructor, if like you are a grad student or a postdoc or early career scientist, usually the applications happen around January or so, February. Then students that participate in these programs, the applications happen around April or so. And volunteers, we try to recruit throughout the year, and depending on what your interests are, what part of the organization you want to get involved with, there might be different opportunities. But yes, definitely send an email, and so that we can learn more about your interest and what stage, career stage you are at.
And my second question is more scientific. I know you're talking about the prediction models for your motor proteins, and you talked about how the ones that you made the mutations in, they were more capable of walking longer.
Mm-hmm.
So I was wondering, like, translationally correlating that to maybe disease. Is there anything that you found that maybe could correlate to some type of disease with motor proteins? Either sorry.
Yeah, thank you. That is a very, very good question. So in the context of diseases associated with motors, actually what the story is a little bit more complicated, because these motors, to be able to walk in, for example, a neuron, like in human cells, usually it's a complex of proteins. So not only dyning, but there is all these regulators. There is LIS1, there is dynactin, there is the story is that in order for building motor so that it can start walking, you need all these different additional proteins. And there are many diseases that are associated with the incorrect assembly of these complexes. So for example, lissencephaly is a disease that is caused by mutations in one of these additional proteins that is actually called LIS1, and that is why the disease is called lissencephaly. So it seems that mutations at the level of these, the foot of this protein, and that modulate the processivity might not be found because the hierarchy is at the level of, okay, maybe sometimes you actually, if you have these other proteins disrupted, then the entire motor becomes dysfunctional and it cannot walk anymore. But what we found, which was very interesting, is that some of these, I mentioned that the wild type dining only in yeast. So because we did some experiments, these particular experiments in yeast, we're only able to walk around 2 microns. But some of the mutations that we did increase the processivity 5 times or 6 times. So we found that some of those mutations are actually the wild type versions of other cell types. So for example, in these filamentous fungi, or in other cells that are not human cells, but like where you need these very long walks, like trafficking from there is a species called Chlamydomonas that has a very long microtubules. The wild type version of those proteins is actually the ones that we found the mutations for in yeast. So there is a correlation, and in nature there might be in some other species where tuning how long these proteins walk is correlates with their function.
Yeah, thank you so much.
Is there, yeah, last minute question before I leave? I'm an early stage PhD student. I want to participate in this opportunity and learn more about detection and support for medical healthcare. Okay, yes, to participate in the science clubs, there is, as I mentioned, there is call for applications usually around January. So stay tuned, visit our websites, and yeah, you can apply, and if you get selected, you can participate in these programs. Yeah, feel free to email me if there is any additional question. And I think there is going to be more instructions, but like there is another session in a few weeks to discuss more details about engineering T cells for specifically target cancer cells while sparing normal tissue, which is a work that I did during my postdoc. So we can discuss more details then.